Assalamu alaikum. Dear students, this is me, Atiq Ahmad. Today we are going to study the topics cytoskeleton and centriole. These are two small topics. We'll start from the cytoskeleton. Cyto means cell. Skeleton means skeleton. Skeleton is basically any hard internal structure which is made of bones and cartilage. For example, in humans there is an internal hard bony skeleton. So cytoskeleton is basically the skeleton of the cell. In the cell there is a small skeleton, a mini skeleton, which is known as cytoskeleton. And just like the skeleton of animals and human beings, this skeleton also supports the cell. Just like in higher animals and in human beings, the skeleton support and as well as provide proper shape to the body. So same is the case here, that cytoskeleton provides proper support and shape to the cell. Cytoskeleton is a cell skeleton. It is just like a network of microfilaments and microtubules, and it is found scattered in the cytoplasm. Microfilaments are small filaments and microtubules are small tubules. So the cell skeleton, the cytoskeleton, is made of small filaments and small tubules, and they are interconnected with each other, forming a network, and that network is found in the cytoplasm. And the function of this cytoskeleton is to provide support and shape to the cell. We will move towards the teaching points. Cytoskeleton, as we have already discussed, is made of microtubules and microfilaments. Now microtubules are made of a protein which is known as tubulin protein, and the function of these microtubules is to hold the shape of the cell, to provide proper shape to the cells. Diagrammatically, it is shown here. This is the whole cell. Now this is a microtubule, these greenish in color. Now these are microfilaments, these light brownish in color. Now remember, what is the in between these purplish color? These are known as intermediate filaments. These are known as intermediate filaments, and in higher classes, in first year, we will study about these intermediate filaments also in detail. So this is the cytoskeleton. So we are moving back to the point, microtubules. So microtubules also make cilia and flagella. Cilia and flagella are small parts of microorganisms. These are small structures which are used for movement purpose. So microtubules are found in cilia, flagella, and another place where it is found. These microtubules are also found in the centrioles, a small organelle which we are going to discuss here in this topic later. Now microfilaments, the second components of the cell skeleton. These are made of actin proteins and are used to change the cell shapes. Actin is another protein. This protein is also found in our muscles, and this protein is also found in the microfilaments, and the function of these microfilaments is to change the cell shapes. Now we'll study the second organelle, the small topic, the centriole. Centrioles are hollow and cylindrical organelles found in the cells of animals and many unicellular organisms. These are hollow from inside, and these are cylindrical organelles, just like small cylinders, thin cylinders. And they are found in the cells of animals and many unicellular organisms, but they are not found in plants. Remember this point for MCQs also, that centrioles are only found in animal cells as well as in the cells of microorganisms and various protist cells. Microorganisms means the protist cells. They are not found in the prokaryotic cells. and they are also not found in the plant cells. They are only found in the animal cells as well as the unicellular organism, means the protist cells. There are two centrioles in each cell, and both are collectively known as centrosomes. Per cell, per animal cells or per protist cell, there will be two centrioles, and both are close to the nucleus, and both are collectively known as centrosome. Now each centriole consists of 9 triplets of microtubules. In each centriole there will be 9 triplets, and each triplet contains 3 tubules. So each centriole contains 9 triplets of microtubules, and one triplet contains 3 tubules, 3 microtubules. So the total number of microtubules per centriole is 27, because there are 9 triplets and one triplet contains 3 microtubules. So 9 multiplied by 3, so it is equal to 27. Now what is the function of centriole? Centrioles are found close to the nucleus. We have already discussed it, and their function is to form spindle fibers during cell division. Whenever the cell division occurs, so these centrioles migrate to the opposite ends of the cell, and from the both ends they will form spindle fibers. Now the spindle fibers help in the chromosome movement during the cell division. So we have studied the cell division briefly in class 8, in higher classes, and in this class 9 we will study the cell division in detail in the 5th unit. During the cell division, the chromosome basically arrange themselves on the center place of the cell. That is called equatorial plate. Now the spindle fibers from both ends attach themselves to the centromere part of the chromosome, and then the two chromosomes which are paired up, those are pulled away from each other. So this function is performed by the microtubules, which pull the two paired chromosomes, and these centrioles are basically made of microtubules. So centriole contains microtubules. Microtubules basically form the spindle fibers, and the spindle fibers play a role in the cell division by pulling apart the chromosome, and they move the chromosome towards the opposite ends. We will have a look at the diagram. Now this is centriole. Now these are 9 triplets: 1, 2, 3, 4, 5, 6, 7, 8, and 9. These are 9 triplets. Each triplet contains 3 tubules: 1, 2, and 3. There are 3 tubules per each triplet. So the total number in this whole centriole is 27. 27 tubules are found in each centriole. Per 2 centriole, the total number will be 54. This is all about today's lecture, and next time inshallah we will meet in another lecture. Take care. Allah Hafiz.